collaborated with the Life Sciences Sector Skill Development Council. So we would like to extend our thanks to them as well. And through them, we got access to the entire academia across country, especially life sciences colleges. So a big thank you to our collaborator, LSSDC. Also, we would like to congratulate all the winners. Like in each webinar, as you know, we are doing a Kindle draw at the end of the webinar. So I would like to congratulate all the winners. And I think all the surprise gifts have been already emailed to them. And only few are in a queue, which will be done subsequently in the next couple of days. So big congratulations to all our Kindle draw winners. You can participate in today's Kindle draw as well. And for that, you need to attend the entire webinar. And towards the end of the talk, all the talks after panel discussion, we'll be doing a quick Kindle draw just to encourage the participants. Also, we did our poster session, a very interesting poster session concluded on 13th November. So in total, we received around 45 poster abstracts and the committee screened them initially and 15 posters were shortlisted. And they actually presented live on 13 November webinar. So now the judges are just working on the results. And in the next webinar on 27th, we'll be announcing the results for that. So there are very exciting prizes. 9 poster winner prizes are kept. So 3 best poster winners for first prize, 3 best poster winners for second and third. So like that, 9 prizes. And as we announced last time, this year onwards, APA has taken another initiative to encourage research in academia, especially to encourage the students. So Professor Saranjit Singh Memor Award has been started from this year, and it will be awarded to one of the academic poster winner, and he will get a trophy, free registrations for next year in-person meeting, APA India meeting, and to and fro economy fair, as well as his accommodation. Everything will be sponsored by APA. Boston International. So we would like to start this just to encourage academic research. So for all the poster presenters who have joined us today, just be there, and within a week's time, the results will be out. So would invite you to join next webinar also. Very quickly for Q&A, while the talk is on, if you have any questions, you can post your questions on the Q&A tab or in a chat. And during the webinar, if we are not able to address your queries, then do write to bostondb21 @gmail.com, and we will reach out to the concerned speaker and get your queries clarified. To participate in the poll, you have to attend the entire webinar. And finally, all these talks, all the 18 webinar talks are going to be uploaded on Boston International YouTube channel, so that it will be available for you to review again. If you want to, any of the webinar, if you want to revisit, you can always do that. So that will be happening after the webinar 18 is over. So in early December, the Boston International YouTube channel will be uploaded with all the webinar talks. Around 60 talks are there. So without further ado, let me quickly invite the session moderator, Dr. Vinod from Luxai Life Sciences. Over to you, Vinod.
Yeah, thanks a lot, Deepak. Am I audible?
Yes, you very much.
Yeah, a very warm welcome. Very warm in the sense, yes, it's very chilly in Hyderabad. A very warm welcome to all of you. Welcome to APA India 2021 virtual conference. Okay, we are into the webinar 17. I can say it's a penultimate, a semi-final webinar. Yes. So under discovery and transformation. So talking about discovery, so new drugs are continually required by the healthcare systems to address the unmet medical needs across diverse therapeutic areas. And the pharmaceutical industries primarily strive to deliver new drugs to the market through the complex activity of drug discovery and development. So development, on the other hand, includes optimization of chemical synthesis and its formulation, toxicology studies in animals, clinical trials, and eventually the regulatory approval. All these procedures are like time-consuming, expensive, and above all, these extremely stringent regulatory requirements. So I think API India is one of the such platform wherein people share their thoughts, their experience, discuss their new approaches, which have the potential to ensure the future sustainability of the pharmaceutical industries. So today in this session, we have four prominent speakers. So the first speaker will be Albert Lee. So who will be talking about 999 elite cryopreserved human hepatocytes. Peter, so who will be talking about the membrane transporters, role in drug discovery and development. Alex, DNA encoding libraries in drug discovery. And lastly, it will be Vikas Trivedi. So who will be talking about the Central Bio Analytical Lab, NC clinical trials, case studies. So with this, having said that, I would like to introduce today's first speaker, Dr. Albert Lee. Dr. Lee is currently President, CEO, and co-founder of In Vitro Admit Laboratories, Colombia. So previously, Dr. Lee was President and CEO of Phase 1 Molecular Toxicology, Santa Fe, New Mexico, from 2002 to 2003. A Chief Scientific Officer of In Vitro Technologies, Maryland, USA, 1995 to 2002. A research professor and director of Surgical Research Institute, Department of Surgery, St. Louis University Medical School from 1993 to 1995. A senior fellow and a director, Liver Biology Department, Monsanto Company, 1982 to 1993. A group leader, Cellular and Genetic Toxicology, Lovelace Toxicology Research Institute from 1979 to 1982. Assistant professor and a research scientist, Cancer Research and Treatment Center and Development of Radiology, University of New Mexico from 1976 to 1979. So Dr. Lee obtained his BSc in 1972 under chemistry from University of Wisconsin and a PhD in 1976 under biomedical sciences from University of Tennessee. So he has got an MBA in 2002 from the University of Maryland. So Dr. Lee has devoted his scientific career in the development and advancement of scientific concepts and in vitro technologies to accurately predict human drug properties, including medical fate, drug-drug interaction potential, and organ-specific toxicology. His research is focused on the development and application of human-based in vitro experimental models, especially primary cultured human hepatocytes, and most recently, enterocytes, in the accurate assessment of human drug properties. Dr. Lee was one of the first scientists to successfully cryopreserve human hepatocytes and enterocytes, intestinal mucosa, to retain properties of freshly isolated cells. Dr. Lee and his colleagues have developed numerous experimental approaches using cryopreserved hepatocytes and enteric system to evaluate human-specific drug properties, including drug metabolism, drug-drug interaction, drug toxicology, and pharmacology. So today, Dr. Lee will be talking about 999 elite cryopreserved human hepatocytes, the recent advancement in human hepatocyte isolation and cryopreservation technology, which has led to the development of 999 elite cryopreserved human hepatocytes. So on behalf of API India Chapter 2021 webinar, I would like to welcome Dr. Lee for his presentation. Dr. Lee, the screen is all yours. Thank you.
Thank you very much for the warm invitation, introduction. It's always my pleasure to be here. Let me share my screen. Is everything okay?
Yes.
Okay. So yeah, it's my pleasure to present again. Thank you, Deepak, for the invitation. I would like to talk about my favorite topic, which is cryopreserved human hepatocytes, especially the 999 elite cryopreserved human hepatocytes. As it already been introduced by our chairman, that drug development is a very challenging endeavor, very important but very challenging. It's difficult to anticipate what human drug properties would be from preclinical studies, right? Because of species difference, and even clinical trial may not give you the safety data because of the low frequency of idiosyncratic drug toxicity. As a result, drug development is extremely expensive, over over $1 billion in over 15 years. And our mission of our company, iVal, and also mission for myself as a scientist, is to accurately predict human drug properties using in vitro human systems. So we'll have human-specific drug properties. We hope that will be useful. And for in vitro system to be useful, they have to retain two specific aspects, which is one, they have to represent the organ that it represents. And second, they have to have properties of the species that the in vitro system represents. So our approach is to develop tools, in vitro systems that everyone can use to study human drug properties. So those are our products. We can also do the study for other laboratories that cannot perform the studies themselves as contract studies. We focus on two major organs, the liver and the intestine. And these two organs are intimately related. And I would like to talk about especially the human hepatocyte technologies, the 999 Elite. We are a company that do lots of research. This is some of our publications. So if any one of you want to have some of our papers, just let us know. We are very proud that we have nearly perfected hepatocyte isolation. We now can prepare hepatocytes that we call 999 Elite hepatocytes because of the high viability, high platability, and high confluency as far as long duration in culture. And this is an example of one of those hepatocytes, one of the lots, cultured for over 40 days, remain confluent all the way through. We can always pool the hepatocytes using a patented procedure that we have. We thaw an individual hepatocyte, we freeze them, and then thaw them again. And we're able to have them platable, just like our 999 Elite hepatocytes, and can stay in culture for over 10 days. The question is, do they work well? I will show you some of the properties. Uptake transport, definitely. We have rifampin inhibitable and concentration saturable uptake of almost all of these hepatocytes. You can see that all of these hepatocytes can uptake pravastatin and can be inhibited by rifampin. P450 induction, a very important property, right? Everyone who does drug development requires to have to assay for P450 induction of the drug candidates. FDA requires 1A2, 2B6, 2A4 induction minimally. If 3A4 induction is observed, then you have to evaluate 2C8, 2C9, 2C19 induction. As a result, you have to have hepatocytes. And also, this is another regulatory requirement. You need to perform studies with 3 donors. So let me show you the results of our hepatocyte in terms of induction. This morphology, beautiful hepatocyte. You can see that we have omeprazole induction of 1A2, both mRNA and activity, phenobarbital induction of 2B6, rifampin induction of 3A4, rifampin induction of 2C8, 2C9, and 2C19, both in mRNA and activity. And almost all the hepatocytes are inducible for 1A2, 2B6, 3A4, 2C8, and 2C9. Over half of the hepatocytes are inducible for 2C19. Pool hepatocytes can also be inducible. You can see that these are the pool donor hepatocytes responsive in 1A2, 2B6, 3A4, 2C8, 2C9, 2C19 induction. Another application of hepatocytes is the evaluation of drug toxicity, liver toxicity, a major problem. And these hepatocytes can be used for routine screening, separate out the non-toxic from the toxic drug candidates for further development. These are examples of the toxic, non-toxic pairs. And then you can also use hepatocytes for different species to ask the question, which animal species is most like human in response to drug toxicity? And on the left-hand side is cyclophosphamide, a lot more toxic in the rodents than human. On the right-hand side is tacrin, a lot more toxic to human and primates than dogs and rodents. You can also treat hepatocytes for a long time, since these hepatocytes can be cultured for a long period. And the longer you treat, the more sensitive is the assay. You can see that beautiful dose response and time-dependent toxicity for almost all the toxic drugs that we have evaluated. We have also developed an assay to identify drugs that we call idiosyncratic severe liver drug toxicity, that we call SDILI, severe drug-induced liver injuries. These are the drugs that would be found to be safe in preclinical and clinical trials, but would go onto the market and cause liver toxicity, leading to death, resulting in withdrawal of the drug or black box warning. So we work with US FDA. They provided us with coded drugs, known to be toxic or non-toxic, but they're coded, sent to us. We performed the assay, and we've developed the ROS-ATP assay. ROS stands for relative oxygen species. ATP represents cellular ATP content. We decided that the ratio is important. And then the AUC, the area under the curve, or the dose response curve, measures the entire dose response relationship. It's the most reproducible endpoint. To make a long story short, after 3 years of research, we've discovered that the endpoint, ROS-ATP, plotting ROS-ATP versus concentration using the AUC allow us to identify toxic the SDILI drugs at over 90%, and as well as identify the non-toxic drugs over 89%. So we can identify drugs, 9 out of 10 of them that induce liver injuries. We can also identify the safe ones, 9 out of 10 times. New application, gene therapy. Gene therapy is a major advance in treatment of diseases. The liver represents a major organ where gene therapy research are ongoing. There are many liver diseases that can be treated with by gene therapy. By transducing hepatocyte with different vectors like adeno-associated virus, AAV, or GalNAc siRNA conjugates. And so we have proposed that hepatocytes can be very useful for gene therapy to allow scientists to understand, do the gene therapy modality actually affect changes of the human gene that they desire? And as well as ask the question, which animal species is more like human in response, right? Just like DMPK and tox studies. That we have shown, I will show you our success using hepatocytes to evaluate efficacy and duration of efficacy of gene therapy modality. We performed a study in collaboration with Amgen, and we evaluated a gene therapy, validation drug candidate siRNA for HPRT1 as a model drug gene therapy drug. You can see that after 1 day of treatment, we're able to knock down 90% of the expression of the target gene, and then just one treatment allowed the target gene to be knocked down for over 33 days. This shows the power of the human hepatocyte for gene therapy studies. Right now, human hepatocytes and animal hepatocytes are used extensively in gene therapy research. Lastly, fatty liver diseases. We genotype human hepatocytes to allow application in fatty liver disease research. And like, for instance, PNPLA3 is a major major determinant of fatty liver disease. The GG genotype results in the fatty liver. And we look at our hepatocyte, and we found the GG genotype on the lot HH1072. Yes.
તમને લિંક મોકલી દઉં ફટાફટ.
I'm sorry. Anyway, and then another gene therapy target is MBOT7, the other gene. In this case, it's for HCV patients. TT genotype is important, And we also have TT genotype in our human hepatocytes. And then another disease, another gene is GCKR, is important for non-alcoholic fatty liver diseases. CT and TT genotypes are important, and we do have hepatocytes with that kind of with both TT and CT genotypes. Our hepatocyte can also form spheroids. For those of you who want to use 3D cultures, these are examples of hepatocytes cultured for multiple days as spheroids. With time, they form this tight spheroid. And many of a lot of these 99 hepatocytes are able to form spheroids. So these hepatocytes can be applied in, these are what I call the fully functional hepatocytes, can be applied towards DMPK, can be applied towards toxicology, towards pharmacology, including gene therapy. Just want to show some of our latest data. We're also, one of the major problem of hepatocytes is that they don't survive. The gene expression, the protein function seems to differentiate with culture. We're trying to overcome that. Just to show some of our latest results. We're able to show that by culturing hepatocytes as confluent culture for a prolonged period of time, everything seems to come back, right? Albumin drops at the beginning, comes back, all the markers for differentiation come back after prolonged culturing, including most of the P450 isoforms, including uptake transporters, and then of course we all know that efflux transporters are expressed more and more with long-read culture. So we believe that we now have hepatocytes that can redifferentiate upon culturing. So we have single donor and multiple donor have approved treatable hepatocytes that we call 999 elite. They can be applied towards uptake, metabolism, inhibition, induction, hepatotoxicity, gene therapy, and maybe possibly as a model for prolonged incubation to allow redifferentiation after culturing. All right, so this is our company, and I've described to you one of our most exciting product, the 999 Elite Hepatocytes. If you have any inquiries about our product, please contact Deepak in India. And then these are the other people who can help you with all your inquiries. With that, thank you very much for your attention.
Thank you, Dr. Lee. And in fact, you have completed almost, I would say, 10 minutes ahead of the slot. So thank you, Dr. Lee, for taking us through the entire this thing. If you can just, Doctor, share, yeah, now your video is there. So over to you, Vinod and Prakash Ravi, to take up the questions for Dr. Lee. Thank you.
Yeah, thank you, Dr. Lee. Thank you for this detailed presentation about 999 Elite. So I have just one question. Means when you do the 3D culturing, so means do you have a standard for this one? Means are there any standards, like for how many days you'll be culturing this one?
Yes, for the 3D culturing, we usually culture for around 5 days. That will allow the spheroid formation. The way we perform to do the culturing is put in around 1,000 to 2,000 hepatocytes in a 96 well plate with the low binding plates that we obtained from other vendors. And they will form spheroid around 5 to 6 days. And they can be cultured for over 30, 40 days.
Oh, 30, 40 days. Okay, good.
Yes.
Okay. And you mentioned about the regulatory norms that we should have at least a 3 donor one, correct?
That's right. That's right. Yes.
And you have got a single and multiple donor also. So means how the single donor will help?
Yeah, I think that okay. Your question is okay. The two questions, right? Number one, you asked if regulatory agencies require induction studies from 3 donors. Yes.
Minimum, yeah. Yes.
Yes, many of them. And then the question is, what you are asking is that single donor hepatocyte results, how important are they, right?
Yes.
Is that is your question?
Exactly.
Yeah, I think that. Yeah, I think that for all research to do with human hepatocytes, after you have made, usually what happens is, well, if you want to publish a paper, for instance, with human hepatocytes, almost most of the time reviewers will ask you to confirm with multiple donors to make sure that your observation is reproducible among different individuals. Now, I think that some people criticize hepatocyte because of the individual variation, but that's a real life situation. When we ask the question, is the drug toxic, or is the drug metabolized or not metabolized? Uh, you have to understand individual variations, which is why it's so important to use multiple donors. First, you develop the assay with a single donor, make sure that you know how to measure your endpoint correctly, and then evaluate the results of multiple donors to ask the question: is it generally, or does it generally occur in all human, or would there be big individual variations? That's very important. One mistake in thinking is that, well, if I use a cell line, that everything is the same. I can do my experiment reproducible all the time. Unfortunately, when you do that, you may have, even if the cell line represent human, it only represent one single human. So you never know, is the result reproducible among different donors?
Okay, thank you. So there is,
yeah, thank you for the questions.
yeah, sorry, there is another question from Dr. Shrikant. So he's telling that every organization has an in-house protocol, their own protocol. But do the regulatory will accept the same, or regulatory has a generic protocol so that everyone has to adopt to that one?
I think that regulatory agencies will look at your results as the way they are. I think that the different protocols, they do not differ that much. You know, in general, everyone, the result they want to achieve is that they want to make sure that you have hepatocytes that are healthy upon culturing, for instance, that they're inducible when they're treated with positive inducers. So the results are important. How do you derive the result? The slight variation protocols are not that important, as long as you have the right controls in the culture. But of course, you know, we are proud of our protocols, and so are other groups. All my friends all my competitors, they're all my friends. And I think that as long as you have healthy hepatocytes that perform well for your endpoint, your results should be useful to allow you to ask a question that am I getting the right information that will reflect human in vivo drug properties?
Thank you. So one last question from me. Actually, 999, you say it's a platable form, right?
Yes.
Okay. And we usually do the metabolism, means metabolism grade assays will be performed with suspension one. So do you mean to say that we can do the metabolism as well as in induction studies with the same hepatocytes?
Oh yeah. I think that you just have to make sure that if you really want to understand how is a compound metabolized in vivo, you have to use hepatocytes that are plated within hours and study metabolism, because the longer you culture hepatocytes, the less active they are, which is why we work so hard to try to come up with ways to culture hepatocytes so they will maintain metabolism. So we now, I showed the data that the hepatocyte activity expression of P450 would drop immediately, but then would come back after 3 days. And that's what we're working on. Very soon, we'll have hepatocyte that will maintain metabolism at a regular in vivo level similar to in vivo, but can be maintained for multiple days. Then you can use those hepatocytes for long-term metabolism studies. Right now, with suspension culture, you can only measure metabolism for 2 to 4 hours. and from 0 to 4 hours. And sometimes you may want to study for longer-term incubations for the slowly metabolized compounds.
Okay, thank you. So another question from Nirbhay Kumar Tiwari. So whether Elite 999 is characterized using 10 positive inducers and 5 non-inducers to determine the threshold cutoff values to qualify any compound as inducer or non-inducer as per new FDA guidelines.
Yeah, I think that we have not done that yet. We have done 99 elite hepatocytes with various inducers, different potencies, so that they can be used for to investigate, to estimate in vivo induction potential using the RIS, relative induction score approach, and that works. That works really well for all the lots. And in terms of the new FDA guideline, I think that we are going to also perform those experiments, and I'm 99% confident that we'll have data that will be very similar to in vivo, based on our studies with RS, right, for the relative induction score, which we use around 7 compounds from low to high induction potential.
Okay, thank you. Another question from Dr. Prakash. So can you brief us about if these hepatocytes are transporter qualified for use to understand their role, for us to understand their role?
Yes. Now, in my presentation, right, one of the very first set of data for uptake transport, all our hepatocytes are active in uptake transport based on prophylactic uptake plus or minus the inhibitor rifampin. Now, we are now exploring efflux transport, but I need to find out whether we have the freedom to operate in terms of the Be Clear method for efflux. And so far, we have not done it just because we don't want to infringe upon the existing patent. But I was told that the patent for efflux transport may be expired. So if anyone knows about this, let me know, because we are very anxious to use our hepatocyte for efflux transport also. And based on just staining for CD/FDA staining for efflux transport, our hepatocytes, when they're confluent, they form the biopockets, and we will see concentration of the fluorescein inside the pocket, showing that they're active in efflux transport.
Okay, thank you. So this 999 Elite comes in a single donor or a 3 donor? That was another question.
Yes, we have 999 Elite represent hepatocyte that we obtained that have that properties, and we have 999 Elite hepatocytes from over 20 donors right now. Practically, almost every single time we make hepatocyte, they are 999 Elite. So we have been that's why we are so proud of our optimization of hepatocyte isolation and cryopreservation.
Sure, thank you.
Mm-hmm.
Deepak, I think there are no more questions.
All right, so then I would like to request you, Vinod, to present a certificate of appreciation to Dr. Lee.
So, Dr. Lee, this is your small token of appreciation. Thank you for the talk, and we would like to see you every time.
Thank you very much. I'm proud
Thanks
of that. I'm very proud to receive the certificate. It's my pleasure. I love India. I would like to come back to India whenever it's possible. So hopefully we'll see you guys in person really soon.
Sure, Dr. Lee. And then let me, on behalf of the organizing committee, extend you invitation Hopefully, we are hoping that next year a conference will happen in person. So we would like to extend the invitation to you for the next year conference, and as the survey goes, I think it may be in Goa. So we'll be more than happy to have you back in India next year in person.
I would love to go to Goa too. Thank you so much.
Thank you, Dr. Lee. And may I request all the panelists to kindly keep your lines muted? Yeah, thank you. And if there are any further questions for Dr. Lee, then feel free to post it in the chat box or a Q&A. And since it's too late for Dr. Lee, so would not hold him any further, and he may not be available for the panel discussion at the end, but we will definitely reach out to Dr. Lee and get your questions answered. So thank you once again, Dr. Lee, for a wonderful talk. Thank you.
Thank you.
Goodbye, everyone.
Thank you. So now may I invite our moderator, Prakash, to take over and let us introduce our next speaker. Thank you.
Yeah, thank you, Deepak, and good morning, everyone, and a very early good morning to you, Peter. So the next speaker would be Dr. Peter Anhert from KU Leuven in Belgium, and he's going to start talking to us about membrane transporters and its role in drug discovery and development. To give you a brief introduction to Dr. Peter. Peter is currently the professor of pharmacokinetics and in chair of the department at pharmaceutical and pharmacological sciences in KU Leuven. He did his D.Pharm and PhD from the same university, and then went to do a postdoc at the University of North Carolina in Chapel Hill, where he worked on development and application of non-clinical models for hepatic drug disposition. Then he came back to Belgium to work with J&J, and then in the preclinical pharmacokinetics department, and then moved on to back to his alma mater, KU Leuven in 2005, where he's currently leading a group on mechanistic pharmacokinetics. His special interests are in the area of ontogeny of ADME relevant proteins, prediction of drug interest cholestasis, and the development and application of IV-IVE techniques related to PBPK modeling. More recently, he's also been working in the area of IMI consortium, aimed at predicting exposure of neonates to maternal medications during lactation. He has published over 148 publications based on PubMed, and his H-index is about 41. He also teaches in KU as a professor. He teaches general pharmacology, pharmacokinetics, drug life cycle, advanced drug development, and drug compounding. Recently, in 2000, about 3-4 years ago, he also started a spin-out company called Bionotis, where he currently serves as the CSO. So it's 5 a.m. in Belgium right now. So thank you very much for getting up so early to talk to us. And over to you, Dr. Peter.
Thank you very much for the nice introduction. And yeah, it's really my pleasure to be here this morning. So I'm very grateful that I have the opportunity to speak to you today and give a presentation. One thing that you didn't know and you weren't able to mention, but I do want to mention here, it's a kind of a special day for me because I'm turning 50 years actually today. So you are the first people alive seeing me and being able to wish me a happy birthday. So it's kind of a special day.
Yeah. So on behalf of the committee, organizing committee, and all the participants, let me wish a very happy birthday to you, Dr. Peter.
Thank you. Thank you. Okay, so let me go ahead and start to share my screen. And then just let me know if you can indeed see my screen.
Yes, we can.
Okay, great. So yeah, so the title of my talk today is membrane transporters and what's the role in drug discovery and development. So just, and this was already mentioned in the beginning, so apart from the fact that I'm a professor at KU Leuven, I'm also involved in Bionotis. That's kind of a short disclosure. So this is what I would like to cover today. So I will talk, of course, a little bit and give you a short overview about the role of drug transporters in the human body. So it's a little bit of background, introductory information. Then after that, I will go give you a few examples about the clinical relevance of drug transporters. And then as a third point, third part of this presentation, I will dive into the in vitro experiments that can be done to study drug transport, different in vitro models that we have used in my lab in Leuven, what kind of probe substrates and diagnostic inhibitors can be used, and what kind of endpoints would be feasible. And then we'll come to conclusions. Initially, I was thinking to maybe also cover in these 30 or 25 minutes PPK modeling, but if you have some questions about it, you can definitely ask me, but it's probably a little bit, too much to cover it also in here today in the same presentation. So to start, as kind of an introduction, I would like to come back to this review article that many of you, I'm sure, are familiar with. It was published in 2013 about transporters in drug development and clinical pharmacology, essentially saying that we have about 400 membrane transporters divided into major superfamilies, the ATP binding cassette family and the solute carrier family, so the ABCs and the SLCs. The most interesting, of course, in this article, so to speak, is this figure where you have an overview of the different organs, or I would probably say more biological barriers that are present in the human body and where transporters are active. Today, I will focus mostly on the liver because it's also my area of expertise, where you can see if you draw very schematically the hepatocytes at their interface, there are bile pockets, bile canaliculi, and you can see it's getting very crowded there, both at the sinusoidal and the canalicular membrane of these hepatocytes. Transporters are mediating the vectorial transport from blood to hepatocyte to bile, and also sometimes also back into blood after, for instance, metabolism has occurred. So the most important transporters, if I have to pick a few, are of course those of the OATP family, especially OATP 1B1 and 1B3 in the liver. Also NTCP is an important one. And also I will cover today, I will tell you a little bit more about in vitro experiments that we did for organic cation transporter 1, OCT1, one of the transporters that is most highly expressed in the human liver. At the canalicular membrane domain, you have the well-known P-glycoprotein encoded by MDR1, but also, for instance, BSEP is a very notorious one, I would say, because it's involved in bile acid export into the bile canaliculi. So just to show you that this is not a theoretical exercise, that transporters are really important, I'm going to cover a few examples. For instance, this one, this article that basically was talking about the impact of SLCO1B1 polymorphism on the pharmacokinetics of pravastatin, multi-dose pravastatin. The most interesting part, again, of this paper is what you see in this figure. If you have pravastatin plasma concentration time profile, what becomes very clear immediately is that the SLCO1B1 variant, and that is shown here in the filled black circles, carriers have more than twofold increased exposure to the SLCO1B1 substrates. It's a synonym for the OATP1B1 substrate pravastatin. So indicating indeed these transporters are directly determining the pharmacokinetics of drugs that are substrates for them. Another example we can give is the one that is shown here, what was published in this paper, actually in 2017. And this was about the interaction that exists between isavuconazole, it's a quite a recent azole antifungal, and then also atorvastatin and other drugs, including metformin. So again, here, this example, and this is another example, of course, and polymorphism, this example in terms of drug-drug interaction to basically to illustrate the importance of the transporters. And so again, if you look at the plasma concentration time profiles of metformin administered alone, and this is the black circles, so the lower curve here, or administered in the presence of isavuconazole, we know that metformin is not significantly metabolized in human. Well, you see a much higher exposure in the presence of isavuconazole, and that indicates the interaction between metformin and isavuconazole at the level of OCT1, again emphasizing the role of transporters in pharmacokinetics. Another interesting one, and then we go back a little bit again in time, published in 2009, is the one here regarding the interaction that exists between, in this case, ritonavir and tipranavir, tipranavir combined on atorvastatin and rosuvastatin. Especially the effect on rosuvastatin is interesting because this is a statin that is not significantly metabolized. So if you do see interaction, you are quite sure this is related to the transporter effects. So, and that's what is shown here in this graph, where we can see in the closed circle, so the higher curve, the higher plasma concentration time profile of rosuvastatin, that indeed this must be due to the interaction of tipranavir, and we also have data that's illustrating the interaction with OATPs combined with ritonavir. Also maybe some interaction at the level of BCRP will be there, but clearly transporters defining the pharmacokinetics of rosuvastatin. And then one of the more famous publications many of you will also be familiar with is the interaction between atorvastatin and rifampicin published by the group of Les Bennett in 2007 already, where you can see a very significant interaction, especially when you look at the plasma concentration time profile of atorvastatin acid, where you can see this huge increase in the exposure to atorvastatin when you combine it with rifampicin, a well-known, if used as a single dose, a well-known inhibitor of the OATP transporters. I just want to draw your attention that this manuscript by now was published over 250 times. So definitely indicating, also emphasizing the importance of this kind of interaction. So I think somebody raised his hand. Is there a question already? If
no,
not,
Peter, you just go ahead and finish the talk and we will take all the questions at the end. Yeah.
Okay, and so then this was published in 2020. What is the clinical relevance and also the regulatory perspective of the organic anion transporting polypeptides? I think the conclusion, if you read through this abstract and through the paper, of course, is that single dose rifampicin, as it was used also many years ago in the study of the group of Bennett, qualifies as a relatively selective OATP1B3 inhibitor, but at the same time, probably we need some novel index inhibitors. If you really want to document, further study these interactions, you need good inhibitors because you need those to be able to find out what is actually the role of these transporters for your drug candidates and what is the extent of interaction that you can expect. Of course, then the other question is, what do you do if you want to verify whether your compound is a transporter inhibitor? So then you would actually have to co-administer it with a substrate. Well, and the good news is that in recent years, it became clear you can also use a biomarker for transporter activity. I'm just giving one example. This was published in 2018 by a group that we also have collaborated with in the past in the Johnson & Johnson DMPK department in Belgium. And this was about the clinical investigation of coproporphyrins as sensitive biomarkers to predict mild but also strong OATP1B1 mediated drug-drug interactions. Again, what you can see here, and now we're just looking at CP1 in panel A and CP3 in panel C as biomarkers for OATP1B1 and 1B3. You can see when there is co-administration of rifampicin, and that's a light gray line up there, that you immediately have a response, a reaction of this biomarker, and you see a very clear increase within a few hours already in the levels. So if you do have a sensitive biological equipment, and most pharmaceutical companies have that today because we are measuring here in the lower nanomolar or even sub-nanomolar range, well, you can very easily quantify or detect whether your compound is potentially an inhibitor. Also, one of the more interesting figures is what you see here. This is actually what we saw for what we did see for, or has been seen for CP1 and CP3 in function of the concentration of the moderate OATP1B1 inhibitor simeprevir, where you can see at a certain concentration, both levels of these CP1 and CP3 will gradually go up in function of concentration. So even for moderate inhibitors, you can sensitively and selectively monitor the inhibitory potency and get very good predictions about the drug-drug interaction potential. So coming back to the more general question, if you want to predict these transporter-mediated drug-drug interactions, where do we want to start? Well, first of all, we need to have in vitro models, and that's what I'm going to cover in the next few slides. We need, for instance, to have transporter-expressing cell line. We can have hepatocyte-based model, and the previous talk by Albert Lee was a very good introduction to that. And how can hepatocytes also be used for transporter-mediated interactions? And then, of course, we do need the right substrates and inhibitors, especially I would say these inhibitors, and this was one of the conclusions of one of the manuscripts that I was also showing. What I will share with you today is one of the studies, or few studies that we did in terms of identification of OTP1B1 and 1B3 inhibitors, but also MRP3 inhibitors. And then, of course, as a third panel, it's also important to have the right endpoints. And I will illustrate to you a little bit how also image-based endpoints can be used, for instance, in hepatocytes to get very quantitative information about drug-drug interaction potential for certain drugs and drug candidates. So essentially, zooming out even a little bit more, what we really need is we need to understand these transporter-related drug-drug interactions and drug disposition processes. We need to understand them from a mechanistic perspective in order to make the right predictions. We indeed read, it was also clear from the previous slide, the right tools, the right compounds and endpoints, and that allows us to make reliable predictions. In order to cover this a bit, to illustrate a little bit more, what can you do? How can you identify inhibitors, for instance, of the OTP1B1 family? This is a paper that we published back in 2013, where we actually combined in vitro and in silico tools to identify novel inhibitors. Essentially what we did, it was quite an easy or a straightforward in vitro model in collaboration with the University of Zurich, Bruno Stieger there. We were able to obtain these Chinese hamster ovary cells overexpressing 1B1 or 1B3. We did use fluorescein as a transporter probe here, which is transported into these cells by this transporter, and then we screened. This was an academic exercise, a compound library of about 2,000 compounds, and we did look at the concentration dependent, at least for some compounds, the concentration dependent effect of the intracellular fluorescence, and could see that for known inhibitors we did have these concentration dependent decrease in the intracellular fluorescence. We were able to rank our 2,000 compounds in function of the sodium fluorescein uptake, and we could identify up to almost 200 compounds that have at least 50% inhibition in this uptake. We also then we were able to develop in collaboration with a group in the Netherlands, an academic group in the Netherlands, University of Leiden. We did identify structural properties that correlated with OTP1B inhibition positively or negatively. And this was also reflected in substructures that would even contribute to inhibition or actually not contribute at all to inhibition, so the opposite. Without going into detail, we also did a prospective validation of the model, which was successful, showing that if we selected I think about 50 compounds and did an in silico prediction, then after an in vitro evaluation, we could confirm the reliability of the model with a reliability of about 80%. So to summarize, by combining in vitro work, and so a little bit of screening, it's not a very large library even, with the development of an in silico model, we were able to contribute to the selection of novel OTP inhibitors, and that on the long term, of course, can also contribute to the use of such compounds in clinical studies. So if I have a little bit more time, I would also like to cover a much more recent study where we didn't look at the uptake transporters, but in this case at the sinusoidal efflux transporters, especially in the liver. Then I mean MRP3, and you see that indeed efflux transporters are not only present at the canalicular membrane, but also at the sinusoidal membrane. And the good news that I mean, what we have also experienced is that the activity of these efflux transporters, as opposed to what you see at the canalicular membrane, the activity of these efflux transporters at the sinusoidal membrane remains intact. So you can use hepatocytes in suspension for sinusoidal efflux transport. This is the probe, the fluorescent probe again that we used. We did this study in rat. I will tell you later why we didn't do this yet in human. So this is carboxydichlorofuranacin diacetate. It's cleaved by intracellular esterases. It traps this fluorescent probe inside your cells, and then you have active efflux by MRP3. So indeed, we were able to set up the same kind of study using CDFDA as a probe that goes in passively and then measure intracellular fluorescin in function of time, and again screened the same library of about 2,000 compounds. We did use the oil spin method. Again, with respect to time, I'm not going to go into detail about the oil spin method, but it allows you to quickly basically separate the extracellular medium from your hepatocytes, and so to measure the uptake after a relatively short incubation time. And what we could see here, and you can see it in the graph here to the right, that if we used a known inhibitor like benzbromarone, we were able to clearly show that, and this is an MRP3 inhibitor, we clearly show that you have a much increased intracellular fluorescence, so indicating that we did have enough dynamic range to go ahead and screen this library of 2,000 compounds. Again, we did the same, more or less the same exercise. We developed also based on this in silico model. We're able to define what are the structural properties correlating or not correlating with MRP3 inhibition in this case. So as kind of conclusions, and for those two examples, we combined in vitro and in silico approaches to identify novel transport inhibitors. Also in the case of OATP, especially also activators were identified. And I think it was relatively novel also that we could use in this context also primary hepatocytes, in this case primary rat hepatocytes, but we have started to do this with human hepatocytes also to study sinusoidal efflux transport. And the fact that enzymes and other transporters are also present there makes this assay more bio-relevant. What we could say by now is that carboxydichlorofluorescein is a poor substrate of human MRP3. Of course, also we have the more limited availability of human hepatocytes, but that has more or less been solved now. So we have been doing experiments with coproporphyrin 1, so one of the biomarker substrates in human MRP3 membrane vesicles, and to further study the role of MRP3-mediated efflux and potential interactions in the human liver. And so then maybe a last question for today is, what else can we do with these hepatocytes? Well, they can also probably be used to study the interdonor variability. And also in the previous presentation, I think this was covered already a little bit. I think this is really an asset of primary hepatocytes that is sometimes maybe not really recognized, that yeah, you can actually use these hepatocyte batches to get an idea about the intersubject variability that exists in certain transporter activities, but of course also in enzymes, but we're talking transporters today. So we have this study published in 2017, where we were able to compare about almost 30 batches of cryopreserved human hepatocytes in terms of OCT1 activity, and also made a comparison with the variability that we could see in the mRNA expression, and even looked at the genotype. To make a long story short, this is the summary results of this manuscript, where you can see the uptake of ASP+, which is a fluorescence probe for OCT1, as many of you probably know. And when you look at the different batches that we screened, you can clearly see you always have to look at the difference between the control condition and then the condition in the open bars in the presence of inhibitors. You can see quite some intersubject variability in this OCT1 activity, which very likely is a reflection also of at least some intersubject variability that will be present in the human population if you compare different patients. And also maybe a bit surprising is that when we try to make a correlation between the uptake of ASP plus and an OCT1 substrate and then against the relative mRNA expression of OCT1, and we did the same for OCT3, but this is the figure for OCT1, we did have a significant correlation, but you can see that the correlation is not too nice. So it really makes sense to also measure activity and not just mRNA expression to get a better idea about this intersubject variability. These are then unpublished data, again, looking at OCT1 activity. So this is an exercise that we did in collaboration with BioIVT, providing us with hepatocytes, human hepatocytes of different, of donors of different ages, going back also to the neonatal range. And where you can see again with, when looking at OCT1 activity using ASP plus as a probe substrate at the neonatal range, you have about 20% activity. So basically we were more or less able to identify the ontogeny profile based on activity using human hepatocytes for OCT1, which has become, of course, a very important transporter by now for disposition at the level of the human liver. So this is a little bit the conclusion, the conclusions that we had there. I'm not maybe going to read it over so that we have a little bit of time also for discussion. I think I just have a few minutes. What else can we do with the transporters? Well, of course, we can also, or what else can we do with the hepatocytes? I wanted to say, well, we can also use them to make cultures. And look at also the canalicular transporters. And so if you want to look at the canalicular transporter activity, you will have to make cultures, for instance, sandwich cultures as one option. And that's the model system where we have experience with. I think many of you will be familiar with it. Essentially, what it means is that these biocanalicular are reestablished in vitro, and you can then actually use, again, fluorescent probes to study the disposition of these probes in this biocanalicularly. Again, here also, we did use the very convenient probe CDFDA. It goes in passively, and then when you have cultures, it will be secreted into these bile pockets by MRP2, which you can, again, easily see here. One of the questions that we had a few years back is, well, can we also use this intracanalicular fluorescence and determine it, quantify it, and then also basically quantify the extent or the size of the interaction between an MRP2 substrate and then an MRP2 inhibitor. Again, to make a long story short, yes, this appeared to be possible. You can define on the images that you take regions of interest, either the canaliculi or the cells and the canaliculi, and then you can actually compare different compounds. These are just a few examples. You have, on the top left, you have the control condition. So we studied different compounds, including saquinavir, lopinavir, tipranavir, and you can see, depending on the protease inhibitor that you study, you have quite a different image. Especially for tipranavir, you almost have a complete reversal of the image. So the biocanalicularly are completely black. So, and this turns out to be a very potent MRP2 inhibitor, both in rat and in human. And this was published back in 2018. We were able indeed to calculate a so-called image-based biliary excretion index. I'm not going to go into detail how this was done. The details are in the paper. But I guess the good news really was that we were able to rank very precisely different protease inhibitors, and we studied almost all of them that are on the market according to their image-based ranking, according to their potency for inhibiting MRP2. And when we compare to the conventional assay that we conducted almost 10 years earlier, well, that we had a much higher precision with this image-based analysis, with the additional advantage that you don't have to consume so many cells actually to demonstrate this interaction. And then I will not cover this into detail, but I think maybe some of you are aware of the fact that we also did do quite some work about the role of bile acids. So there are also transporter substrates and how interactions with bile acid transporters in the liver can contribute to drug-induced cholestasis, and how you can use in vitro models like cultured hepatocytes to better understand this. This is work that was started almost 10 years ago together with one of my former PhD students, and I think many of you will know him, with Sagnik. Then more recently in 2018, we published this review article together about drug-induced cholestasis. What are the mechanisms, models, and markers? again, I definitely invite you to read through some of the papers that we have published together on this topic. Probably one of the more interesting one to start with was the one published in 2014, where we basically established this cultured hepatocytes as an in vitro model for the assessment of drug-induced cholestasis. And then more recently, last year, and this was a collaboration with several former PhD students in my group, basically refined this assay a little bit, and this was published in Methods X, so in 2020. And then earlier this year, actually, we further refined our assay, not only looking at the toxicity of the compounds in the presence and absence of bile salts, but also looking at the bile acid profiling. And we did do this exercise for bosentan in sandwich cultured hepatocytes. Okay, so with that, I would really like to conclude and thank all the people, all the PhD students that have done the work, former PhD students in my group, and the ones in bold are the ones that contributed to some of the data I've presented today. Also the current PhD students in my group, on which I can rely for new data. Also our collaborations, both in companies and in academic context. And with that, the floor is open for questions.
Peter, if you can stop sharing, we can display a video then, yeah.
Yeah.
Over to you, Prakash, to take the further questions.
Yeah, thank you very much, Peter, for coming this early in the morning on your birthday to talk to us. So thank you again for that.
It's my pleasure.
So I'll go with the questions that were already, that's on the chat box. So this question is from Anup. He's asking you, can you comment about the decision tree about the predominance of polymorphism and its impact on functional activity of transporter?
So the decision tree, so then, yeah, that's a little bit referring back to, I guess, to the regulatory requirements. I think, well, for every study there, it's important to make sure that you provide a fit for purpose answer. So to what extent is an interaction really, or is the impact of a polymorphism really a problem? I think it also depends on the therapeutic window of your compound, whether it's going to be really a problem. So I think it's a little bit hard to really give a very general response to that, and it really depends on the drug that you're looking at and how important is that transporter, then how much does it contribute to the pharmacokinetics of your compound. If it's an individual transporter and it's the only pathway for your disposition, then the impact can be quite significant.
Okay, so another question from Anup also is that should we be more concerned about polymorphism of uptake transporters rather than efflux transporters, or vice versa?
I think, again, I would be potentially concerned in both cases. And again, like I said, you need to understand the mechanisms and what is the exact role of the uptake and efflux transporters. To what extent do they keep each other in balance? And also, let's not forget, we should not only look at the plasma levels, but also what is happening at the intracellular level. So for instance, in the case of bosentan, we know you have an accumulation ratio for the unbound bosentan of 34. So that has been determined previously. So that means if you have some changes in these intracellular levels, it can have an effect on the toxicity profile. So again, here, let's try to understand the mechanisms, and then we can define how important they are for a certain drug candidate or even a marketed drug.
Thank you. So I have one question from my side. So how would you work? What type of models would you use to understand renal transporters? Because you've been talking primarily about hepatic transporters. So what type of systems, or what would you be using same kind of methods for your renal transporter analysis?
For renal transports, it's of course not my area of expertise, but I think, there are several groups. I don't know the name now, but several groups also in the Netherlands, for instance, that I know are very experienced with it, also using organoids. But I think with cell lines expressing the relevant transporters that are involved in the disposition of your compound, that's a very good start. Huh? And if we do know what are the major transporters, the major transporters contributing the disposition to renal disposition. If you want to specifically study those, you can also use membrane vesicles. Why not? Also, membrane vesicles are a very great model system to directly study the interaction. And also this kind of data can be used then later, for instance, in PBPK models to predict what may be going on at the in vivo level, because eventually that's what we want to do.
Okay, so I don't see any further questions in the chat box. Is there anyone in the panelists would like to ask any specific questions online? I would, we have a couple of more minutes before we close.
Maybe Prakash, I think they may post questions while the other talk is on. So why don't we present a token of appreciation as a certificate too? Yeah.
Okay, sure. So on behalf of APA India, I would like to definitely present this certificate of appreciation to Peter for his excellent talk on membrane transporters and role in drug discovery and development. I think it gives a lot of people at least what they need to be looking for to understand where they will go forward from it. So thanks a lot to be up on such early in the morning, and we would like you to continue to stay online if you're possible for us, in case there are some more questions that we can take up in the panel discussion, which probably take place another hour or so. Thank you.
Yeah, I will try to do that. And thanks again. It was really a pleasure to be here.
Thank you, Peter, for a wonderful talk. And very quickly, would like to extend our invitation to you. Hopefully next year when we do in-person meeting, we would be more than happy to have you in person delivering the lecture on transporters. So
I'm already looking forward to it.
And also we will have to take this small party from all of from you on behalf of all the participants because of your birthday today. So next year we may have one party from Peter. So thank you.
Yes. Yeah. Very good. Yeah.
Thank you. Thank you. So Prakash, over to you. Let's move on with the.
Yeah. So our next speaker in this series is going to be Dr. Alex Satz from WuXi AppTech in the US. So Alex has got over 15 years of experience in building DNA encoded library technology platforms and is currently the Senior Director for Strategy and Develop Operations in WuXi AppTech. Prior to joining WuXi, he built and led the DNA encoded library platform with Roche at Basel in Switzerland and Nutley, USA, and also helped to develop the first industrial scale DNA encoded library platform at Praxis Pharmaceuticals, later which was pursued by GSK. He worked as a medicinal chemist at AstraZeneca. He was an NIH postdoctoral fellow at Harvard Medical School and earned his PhD from UC Santa Barbara. And in the field of DEL, he has authored more than 18 peer-reviewed articles and 2 book chapters. So today, Alex is going to talk to us about DNA encoded libraries in drug discovery. Over to you, Alex.
Hello, good morning. Let me just make a quick check and make sure that you can actually hear me. That's always important.
Yeah, we can hear you. And we can see you too.
Perfect. Good. Okay, let me share my screen, or at least share my PowerPoint presentation.
Yeah, we can see that.
Okay, great. And let me get my laser pointer. Okay, well, thank you for the introduction, and thank you for all of you who have signed in on your Saturday afternoon or morning in order to listen to these presentations. As said, I'm going to talk to you about what we at WuXi do regarding DEL. So that's a relatively new way of finding hits. So let me talk about this plot that I have here and what it means. So along the x-axis, we have the year, and the y-axis shows the number of papers published in the DEL field over time. And I entered this field in about 2004. And as you can see, back then, there was very little work being done. So we were really one of the first groups who entered this field. But as you can see now in 2021, there's a lot of work being done in the DEL field. And indeed, this has gotten to the point where really every single big pharma uses DEL in order to find hits for the programs that they work on. So let me just give you a few examples of published cases of hits that were found and the different target types. Because it is sometimes asked when you do run a Dell screen, what kind of hits will you find and what will those hits look like? I show some examples later on, but here's just a few. All right, so what does a library member in a Dell actually look like? And I'm showing you a very simple scheme here. So there's a barcode, and you can sequence this code. And from the sequence of the code, you can tell what the structure is that is attached to it. And although here it's shown as a rather short, rigid linker, it's actually a very long, flexible one. So the key to understanding the use of DELs and how they're actually screened, it's really best to know a little bit about how these DELs are actually made. So they're made by using split and pool CombiChem. So let me just go through what that means. And then I think everyone will have a better idea about how these DELs are made, of course, and how they're used. So let's start off by envisioning that you have a mixture of 3 different things as shown here. And at any point in time, you could sequence the code. And from the sequence of the code, you could then know what the structure was that was attached to it. And you take this mix of 3 things and you split it amongst 3 different tubes. So that's the split step. And in this case, we would say that the split size was a size of 3. Okay, so now we have 3 different tubes, and each tube is the same exact thing. And we add a synthon, which is shown here, to do a reaction on this side, which is added here. And we do a ligation of a new tag, which is shown here, which adds on to the end of the code, which is shown here. And we do this in 3 different tubes with 3 different tags and 3 different synthons. And in the end, we create 9 new structures, and each structure is encoded by a unique code. We can now take all 9 new structures and we can combine them into a single tube. So that's the pooling step. So we've done a split and then a pool. Now, the difference between what I've just shown here and what we do in the real world is that in this case, the split size was 3, but in the real world, our split size would be something more like 3,000. And so if you do multiple cycles of CombiChem split and pool, a single person can create millions of new structures, and they can do this in about the span of 1 month. So you can make many new things at a very low price, and that is where the value of Dell really shines. All right, so how is the screen itself done with these mixtures of DELs? And the answer is you take the target that you want to screen and you attach it to a solid phase resin. You can do this with a his tag or anything along those lines. You then take your mix, which is your DEL, and you add it. Now, most library members don't have a structure which can bind to the binding pocket because there's not a good fit or a good match. So all these things end up being washed out. What you're left with was what was bound, and you collect those and you then add it to fresh protein a second time, and you go through this cycle a second and a third time. Then when those cycles are done, you amplify the barcode, you sequence them, and from that, you know what the structure was of the hit that interacted with the binding pocket. So that's the basics of how this Dell screen is done. So what do the results of these Dell screens look like? I'm showing you a few cases here of pharma, which has run a Dell screen, and I'm showing you what the target was, what the hit was that came out of the Dell screen, and what the hit looked like after the medchem was done, and what the status is. So for instance, in the case of RIP1, I believe this hit is now in phase 2. And in the case of SCH, the structure shown here is now, I believe it's in phase 1, maybe in phase 2. So those are just a few cases so that you can get in a view of what does a DEL hit look like when it comes directly out of a DEL screen. And of course, what does it look like after the med chemists are done working on it. So it's true that Big Pharma has invested a lot in this, and they have found ways to find hits against the targets that they want to find hits for. But the question is often asked, if you're not in Big Pharma, how do you access these DEL screens? If you're an academic lab or if you're a smaller startup. And in order to solve this, what we at Wuxi have done is we have created these self-service kits. So of course, we still do it the kind of the older fashioned way where you send the target to us and we would do the screen for you. But what's nice about the self-service kit is that we send the kit to you and then you can run the DEL screen in your own lab. Now that has two nice things about it. One thing is that the target you screened, we at Wuxi don't know what it is. So if you want to keep your target a secret, you can do that. The second thing is, of course, it lowers the cost because the physical screen is done in your own lab. And so we're able to offer what we call a DEL Open Kit for academic labs. And this kit is completely free. So let me talk about what that means on the next slide and how that works. First, I'm just going to show you a picture of what a DEL kit actually looks like because it's pretty simple. We have 4 tubes, and in each one of these tubes is a mixture of about 30 to 60 DELs. And that means that there's about 15 billion different structures in each one of these tubes. And so since you have 4 tubes, you can screen 4 different conditions all at once. Okay, so how does actually getting this kit work and screening this kit work? So let's talk about the DEL Open case first, which is free for academic labs. So the circle shown in blue, that's what we do. And the circle shown in yellow, that is what you would do in your lab. So first of all, we make these kits, but then you can go online and you can order it. And like I said, there is no cost except for, I think, the shipment back and forth. So you then run the physical screen in your own lab. It's a pretty straightforward thing to do. And we provide you with notes on how to actually do this. When the physical screen is done, you send the samples back to us. And we do the PCR and the sequencing, and we then convert the codes to physical structures. And we then share all of the data with you, except for the structures. We don't show you those. You would then look at the data, and you would make the choice about what the 5 best hits are. We would then synthesize those hits. And we would send the hits to you, but you still don't know what those structures are. But you can then test them in your own lab, and you can make a decision about whether these hits were good or not. Now, if you feel that these hits are indeed good, you can ask us for the structures of those hits, and we will give you those structures at no cost. And with those hits, you are free to sell them, publish a paper, a patent, whatever you might want. And there's no cost and there's no reach through claim. So you are free to work on those hits. However, DEL Open is overseen by an academic group. So we at Wuxi, we make these kits, but there's an academic group who makes the decisions about how this all works. And it's all about being open. And so in exchange for the structures of the hits, we would ask that you share the target name with us, and the metadata of the screen itself would be shared with the DEL Open members on a website so that others could then go look at that data and possibly work with you in order to make future publications, because of course, the goal here, this academic group, is to publish more. I should also point out that when the compounds are synthesized, we do charge you for that, but we have worked out a deal with the DEL Open group, and it is at a relatively low price. So other than having the hits remade and the shipping, the entire thing is completely free. Okay, so Delight is a little bit different. Delight is really made to discover drugs, and it's made for pharma, both big and small. So how this works is, again, we have a kit, but you need to go online and order the kit, and the kit does have an upfront cost. Again, the kit is shipped to you in your lab, and where you physically do the screen, and you send the samples back to us, and we PCR amplify and we sequence, and then we convert those sequences to the structures. We then share all of the data with you except for the structures themselves, and you decide, did the screen work? And if you decided, yes, the screen worked, then you can pay an additional fee and you can unblind the entire data set. Now, once you have the entire set with all of the structures, you are then free to follow those up in any way that you see fit. Again, there are no reach-through claims from us. There are no milestones. You are free to discover drugs, write patents, and other things. Okay, so those are the basics of the self-service kits, Dell Open and the Delight kit. So let me talk a little bit about the most recent edition of these kits and what they look like. So for instance, the 4th generation Delight kit now has 58 DELs and a diversity of about 10 billion structures in each tube. And the Dell Open kit has 27 DELs and about 4.2 billion structures in each tube. We've just made this 4th generation kit, so it's a little bit different from the 3rd version. So for instance, there were not too many 2-cycle DELs in the 3rd generation kit, but there are many more in the most recent kit. What that means is that we've made many more DELs where the potential hits from these DELs are going to fit better into that drug-like space, meeting the rule of 4 and meeting the rule of 5. To try to show what that means a little bit, we have this PCA plot, and every dot represents one of the structures from the 4th generation kit, and those are all shown in blue. And all of the structures from the older kit are shown in green as these little green dots. And the one thing that stands out about this PCA plot is that we have this new chemical space, which is being filled. You can see it here and here and here. It's only shown by hits or potential hits that are contained within the 4th generation kit. And what are these structures? They are structures that are more lead-like, they are more drug-like, and they now exist in the new kit, but they did not exist in the older kit. And that's why in this PCA space plot, we're seeing them separated out from this main blob here. So as a chemist, trying to interpret what a PCA plot can be, means can be a little tough. So what I've done here is I've shown a couple of cases of structures or DELs that we've made that have gone into the most recent kit. So for instance, this is one example of a DEL formed, about 20 million structures, and 80% of these potential hits would fit Weber's rules for drug-like likeness. Here's another case of a 2-cycle DEL. And in this case, 65% of these structures meet the rule of 5. So these are the kinds of new DELs that we've put into this new kit, and you can see where the chemical space was being separated out from the 3rd generation kit. These are the distribution plots, comparing the 3rd generation kit shown in red with the 4th generation kit here shown in blue. Just for an instance here, if you look at the heavy atom count, you can see that there's a shift on average going from about 39 heavy atoms to, let's say, about 35. So we have a 4 heavy atom shift. And again, we see a shift to the left for the size of these hits, heavy atom count. For the fraction sp3, we're seeing a shift to the right. For the rotatable bonds, we're seeing a substantial shift to the left. So these new structures, in addition to being smaller, are much more rigid. And we also see that for PSA, we see a shift to the left, and for hydrogen bond donors. So all in all, this 4th generation kit has many more drug-like hits in it. All right, so DEL screening generates a lot of data, and there's always this question of, do we really do the best job that we can in order to look at this huge data set and to be able to figure out which hits we should follow up, and are we finding the best hits that the data really tell us about. So what we've done in order to try to address this is we have set up a way to do deep learning on these data sets. So just to put this simply, we run a screen, we have this huge data set, we run the deep learning, and then we take a classifier from that and we look at the commercial sets of anything that we could purchase off of a shelf, and we make a determination about which of those is most likely to provide us with active hits. So let's see if this machine learning effort can actually work for us using the DEL data. So we'll take a target, Aurora A, which has been around for quite some time, and we'll use this as simply our POC. Right, so we take it and we run a normal DEL screen on it using a DeLite kit. And along the x-axis here, we have each of the different DELs that we have screened. And along the y-axis, we see the enrichment levels of the potential hits from each one of these DELs. So each dot here represents a potential structure, which could be a hit against Aurora A, which is the screen that we've run. We more or less randomly choose 4 different hits from 4 different DELs. And we are showing those structures of each of those hits here and what their IC50 curves look like. In this case, all 4 hits were indeed active against, we see one example here, which is probably one of the more unique looking hits that came out of this screen. And as you can see, it is low nanomolar active. And we can also see that some of these hits are fairly small. But with that said, probably the most structurally interesting hit, which is shown here, is a little bit on the large side, so it doesn't quite meet the rule of 5. All right, so that's a normal DeLite screen and how we worked up that data. All right, so now let's look at what occurs when we do the deep learning on this data set and we go into the commercial data and we try to find new hits. So as you can see, here's a case of a couple of structures that we have found. I would point out that we have purposefully only looked for things where the compounds would be rather small, so they would meet the rule of 4. So the molecular weight would be less than 400 and the C-log P would be less than 4. And indeed, both of these hits are indeed active and they bind to the target. Another important point here is that since these are from a commercial source, the price for accessing these hits is quite low. So these hits cost roughly about $100 each. So this indeed is a method that we can use in order to ensure that we're finding hits that fit more into that actual lead-like space, not even drug-like, but in lead-like space. And they, of course, cost $100 each instead of what is the higher cost as shown on the prior slide in order to synthesize a novel hit completely from scratch. All right, so I don't have too much time left, but I'll try to quickly talk about something else that we do, which is to make covalent DELs so that we can find covalent hits. So as you can see here, the interest in finding covalent hits has gone up dramatically in the last 10 to 20 years. This is the number of papers published over time. And as you can see, there were a few published back in the 1990s, and today there are many more published every single year. So what we decided to do was to run a proof of concept screen against BTK, which is a target which has been around for quite some time in this covalent space. And BTK does indeed have a cysteine, which is in its ATP binding pocket. So we will attempt to target it. I would also point out that BTK does have other cysteines as well. And so that's something that we have to think about. We, of course, want to target the specific cysteine and the ATP binding pocket and not simply react with the protein all over. So we've made a large number of different DELs. These are 51 DELs containing 623 million different structures, and every different structure has a covalent warhead on it. We then screen this DEL against BTK and RPOC, and we find that we see one of these DELs gives the strongest enrichment signal. We look at two of these structures, which we are calling compounds 1 and 2. Compounds 1 and 2 differ only by a single CH2 group, and their molecular weight is 366 and 380 grams per mole. So these hits, these potential hits, are very small. We then do a mass spec test in order to determine that these potential hits do indeed react with BTK, and that they react in a 1-to-1 method. So they're not reacting with every cysteine, they're only reacting with a single cysteine, and the interaction is indeed a covalent one. And lastly, we run an IC50 in order to determine if these hits are indeed active, and they are. For instance, after a pre-incubation time of 3 hours, compound 1 is about 100 femtomole or 100 picomolar hit, and compound 2 is about 50 nanomolar. So these are indeed extremely small and extremely potent hits. And although I can't show you their structures, structurally they are distinct from other known hits against BTK. Okay, with I think I have 1 minute left. I will just say that we did do another POC against 3CL Pro, which is from SARS-CoV-2. And just to point out here that these hits were indeed active as well. Here we're showing their IC50s against a range of different 3CL Pros from different COVID strains. And as you can see, in some cases, certain hits are active against some versions of SARS-2, and they're not active against other versions of the 3CL Pro. That's a good sign in terms of we are able to find hits which are selective against some of the targets and not active against others. Okay, so with that said, I want to thank the group who did the work in order to find this data. Of course, I'd like to thank all of you for your attention on your Saturday. And I will be around for the panel discussion, which is going to take place in about 30 minutes. So I can answer any questions then if there's no time now.
Thanks a lot, Alex, for an excellent talk and for walking us through these DNA encoded libraries. So let me see if there are any specific questions. Yeah, I have one question to you, is these targets that you are looking at, are they going to be membrane-bound targets, or could be any kind of targets? Could they be like soluble, or they have to be, can be anything?
Membrane-bound targets are tough. Yeah. So we do need them to be a soluble target. However, some membrane targets can fit that description. But at the moment, although there is a lot of work done on the non-soluble types, I would say that the membrane targets are tough. Yeah.
Okay. All right. Thank you.
Sure.
All right, Prakash. So Alex is going to be there for the panel discussion as well. So we would like to. Would you like to present a certificate appreciation to Alex?
Yeah.
Thank you.
Yeah. So thank you, Alex, for an excellent talk on the DNA encoded libraries, especially in early drug discovery, and how WuXi is helping us generate these hits at fairly low cost, even especially in case of academics. So thank you very much for your excellent talk, and looking forward to you staying over for the panel discussion where there'll be a few more questions there.
Okay, thank you. Bye.
Thank you, Alex, for a wonderful talk. And just hang on there after this last talk. Again, we'll connect in a panel discussion. Thank you, Alex. May I now invite Srinivas to introduce our next speaker and take the proceedings further?
Sure. Hope I am audible.
Yes, very much. You're audible.
Yeah.
Please.
So going with the theme and bringing in another aspect to the today's session, we have next speaker Vikas Trivedi from Vida Clinical Research India. So Vikas actually has been in biomedical sciences for a while. He is associated with Vida Clinical Research and serves as a scientific and technical lead for biopharmaceutical companies. He plays a key role in method development, validation, and applying these methods to comply with the scientific and regulatory requirements. He brings with him 15 plus years of professional research experience in regulated bioanalysis and has been responsible for developing, validating, and using these bioanalytical methods to support clinical studies for generic products as well as NCEs. So his expertise involves in developing the methods for NCEs, endogenous molecules, chiral compounds, molecules requiring sub-picogram sensitivities, even the liposomal products, small peptides, tissue sample analysis, protein binding studies, also supporting microsampling, capillary as a sample collection technique for clinical studies, and large molecule bioanalysis, especially by LCM-SMS techniques. So Vikas has many publications to his credit, which have been published in peer-reviewed journals such as Bioanalysis, Journal of Chromatography B, Journal of Chromatography Sciences, and so on and so forth. So I request Vikas to come on the stage now and discuss on the topic about Central Bioanalytical Laboratory, the case studies for NC clinical studies. Vikas, over to you.
Yeah, thank you. Thank you, Dr. Srinivas, for a wonderful introduction. First of all, I would like to say good morning, good afternoon, good evening, whenever you are, wherever you are. So first, let me share my screen. My slide is visible, Srinivas?
Yes.
Thank you.
You can put it in the presentation mode.
Yeah, sure.
Yeah.
Yeah. Okay. Once again, thank you all. So today I'm going to talk about specifically for the what are the practical challenges faced by the biotical scientists for the development of I can say the new chemical entities where there is no any reported literature, and I can say the no any product specific guidance are available in a public domain, and I can say only the few information available before initiation of the any development in a biotical for specifically for a new chemical entity. And also I would like to share our experience and touch upon I can say the two case study where the fully validated method and the rugged method doesn't work, while slightly modification in the existing validated method, or I can say the transferring the method from one lab to another lab, or difference, or try to compare the results between the two validated method while applying to assay with respect to the real study sample analysis. So this will be the flow of my presentation. First, I will discuss on the VIDAS as a central bioanalytical lab and role of bioanalysis in the pharmacokinetic study, and challenges like logistic challenge in timeline challenges, practical challenges in bioanalysis of NC, and case studies of NC in bioanalysis. So VIDA is a central bioanalytical lab servicing delivery with the wide variety of matrices like plasma, whole blood, urine, skin, colon tissue, and as a VIDA analyzed 200 case samples and coordinate with more than 200 sites across the globe. Specifically, our bioanalytical lab have a vast molecular experience for the different NC and have more than 69 validated method with the turnaround time of less than 4 days for specifically for the safety analysis for dose escalation study. VIDA familiarly and complies with the regulatory requirement of the US FDA, EMEA, NVISA, and other agency, and enable our client to have a complete confidence in the quality system and the reliability of the data. Also, we have a latest instrument, multiple equipments and technology to ensure the project completion within the quick time frame. And also, we have Watson LIMS, means Laboratory Information Management System for all biological data management system. Also, the data transfer as per the sponsor specific requirement with dedicated experienced project manager team, transparency and open communication, direct management, supervision, and customer centric approach can be virtually an extended of their own team and lab to our sponsor. And we have a dedicated team for a central lab services, which includes project manager, dedicated project manager for NC program, sample management team like custodian to handle the samples while receiving or while clearance from the custom. And we also support for the kits and logistic coordinate with the different site for NC program. And analytical team like PK analysis based on the project, and dedicated Watson team for the NC. This is just a sponsor global pharma companies type of study, NC multi-site. Total more than 40 studies currently are ongoing from the multiple sites. 2,000 samples per year we are doing in a bioanalysis for NC molecule. Service provides sample management, receipt, login, and in LIMS reconciliation before the analysis and after the analysis. And yes, definitely for method development and validation of NC molecules. For specifically for a safety analysis, sample receipt to analysis within 5 days. And we also support for the specific sponsor specific report format with the ECTD. As I mentioned earlier, more than 69 validated method which we have at present for NC molecule. Exploratory studies, for example, like skin tissue, plasma protein binding, chiral impurity, and microsampling for like CAP and TAP device. Also, we have a validated, and I can say the partially validated method for the exploratory analysis studies. Then role of bioanalysis in pharmacokinetic studies. So just I would like to touch upon some part of the basic of the pharmacokinetic analysis. So pharmacokinetic analysis is an experiment trial, determines the theory how the new drug react with the human body. This study provides critical information about the new drug. Pharmacokinetic refer to the movement of drug through the body, and PK describe the like characterization, ADME like absorption, distribution, viability, metabolism, excretion as a function of time. The PK measurement first in human study provide a tentative confirmation of safety and exposure from the tested dose level. When the PK information is needed, dose escalation, a common practice is to be performed interim PK analysis progress, where the PK is examined in one group before proceeding to the next higher dose level as a part of safety measurement. So this is, I can say, the importance of the bioarticle for PK sampling in the different stages of clinical trial. From, I can say, from drug discovery, preclinical research, IND submission, and from phase 1 to phase 4 and the post-marketing. In each and every stage, there is a very and crucial important role of the PK analysis, or I can say the validated bioarticle method. So the role of bioanalysis in the pharmaceutical drug development is classified, discussed with the focus activity that are performed within each stage of clinical trial. So bioanalytical technique continue throughout the preclinical, clinical drug development, human studies, I can say multi-central studies in target population, drug-drug interaction study, bioavailability studies with the different formulation, effect or different type of formulation, or into post-marketing support and may sometimes a therapeutic drug monitor. The application of pharmacokinetic, the maximum information can be extracted from the data. Subjects are available as in first in human clinical studies, and then this information is applied and interpreted of the next study during the drug development phase. So PK sample analysis require careful planning with the logistic between the clinical conducting and within the laboratory as a central bioanalytical lab. The laboratory analyzes the sample and the PK calculation. So bioanalysis is the quantitative determination of drug and their metabolite in biological fluid like serum, plasma, urine, fecal, in some cases cerebrospinal fluid and aqueous humor. So challenges of the central biological lab. So there are two kind of challenges which we can say, like logistic and timeline challenges and practical challenges in bioanalysis of NC molecule development. So for logistic and timeline challenges, the first challenges is reference standard management. I can say for application of the import license and approval from the local agency and clearance from the logistic at India. So as per the recent notification received from the OECD, now the import license for working in reference standard is not required. So other only licenses and the approval process is required. And for same, we have a dedicated team to communicate with the custom and the agencies. And also we know that accountability of reference standard is the main thing, and also the expiry of the NC molecule is short expiries due to limited stability data information for this NC molecules, and also the quantity for some of the metabolite is less. So we have to manage the things with the less quantity for the quantitation of specifically for metabolites or evolution of metabolite. Then sample management. For sample management, declaration of safety and integrity during transit need to be maintained. Then import to coordination from the site, custom clearance, logistic in India, and biological sample import permit for testing purposes. Right now it is not required, but again we need a dedicated team to dealing with the sites and site coordinators for the timely manner receival of the samples. Then last, sample results timeline. For I can say for first in a human study, the timeline are critical to share the PK data along with the safety data review for the further decision on trial within 4 to 5 days. In case of more sample, we need to add more equipment or multiple equipment. So it should be have a similar kind of setup with the same equipments to achieve the final timeline. Then fast switching and the decision making is required. So dedicated scientific team is required to take a call on the method or in between the method. Sometimes the predicted the rain doesn't work, then immediately we need to take a call and switch to method with and revalidate the method based on the existing dose requirement. Then faster communication is also required. Is a key for any successful trial. So PK scientists should revert the trial lead on the immediate basis or based on the PK results and communication with the project manager and the coordinators. For safety cases, some communication are critical for a trial as this is affect the progress and the decision of specific NC molecules. Data flow. The generated PK data should be reliable, acceptable as a global quality, longer study duration, and reliability is a key concern for any follow-up trials and the decision making. Any mistake or any misinterpretation based on the biological method performance should be communicated immediately to take a further decision, or it may lead to the wrong trial decision. So the importance of the rugged and the reliable biological method should be there. Again, this is the technical challenges. First, we know that method development for NC mogram, as I mentioned earlier, we have a limited method development information with an expected metabolite is feasible, should be part of development program whenever it is possible to get the metabolites, and rugged and reproducible method for NC program, and dynamic linearity range. As we know that we need to cover from the lower dose to the higher dose. So the dynamic linearity range is required to cover all the proposed dose. For example, 2000 times linearity or higher. While developing the method with the higher linearity range, we need to ensure the performance of the method, or we need to select the proper chromatography that should not, saturation should not occur. And also we need to select the proper chromatographic condition and the extraction procedure for an NC program. And as we know that linearity range is dynamic, then we obviously face some challenges like carryover issue and the other issue. So that also should be ensured in the NC program. I think in one of the presentation by Dr. Amarnath Jaswal, they have proposed one mitigating So in few cases or in few NC program, if required, then we can add mitigating blend after the higher standards. If the carryover is not resolvable, then higher concentration needs a dilution like 10 times, 100 times, 1,000 times fold. So method should be that much accurate that after dilution, all the data should be reliable. Then we know that we need to develop for NC molecule with the less processing volume, or I can say the plasma volume for analysis due to limited blood withdrawal in the patient population. And also other determination is required in NC development program like PD determination, biomarker, and safety. So we always develop with the method with the lower processing volume. In few application, if any pediatric studies are planned, like in that case, such like a microsampling device technique, DBS card, tape, or cap device is recommended to use for the pediatric, where the sample volume is very less. Yes, there is a metabolite determination, method development for active and inactive metabolites. So at the time of method development, whether the metabolite is pharmacologically active or not active, but we need to ensure at the time of, I can say, development or validation, or in some cases, we need to ensure at the time of study sample analysis whether any active or inactive metabolite are affecting the assay. Exploratory estimation of active and inactive metabolite in case of specific requirement. Determine the level of concentration and correlation with the metabolism. Then method reproducibility. We all know that for any NC program, the NC program takes around 4 to 5 year 5-year time from the, I can say, from phase 1 to phase 4. So in each and every case, method should be reproducible up to 4 to 5 years. And if required, then we can do some minor modification in the method based on the results of the different trials and the population. Sometimes there is no need to involve the multiple lab in same trial. So cross validation mandatory to present the data reliability between the labs. And also long-term stability data is required for the approximate 3 to 5 year. Method activation frequency. Method should be active and frequently tested to performance to avoid the any misinterpretation of the data and due to criticality of the samples. So before going ahead with the study sample analysis, always perform the method qualification or one precision batch before conduction of the study. So now I would like to discuss about the case study in bioanalysis. So first case study. Method was developed alone for a drug and fully validated. Then all the stability experiment was performed and found well within the acceptance criteria. The study sample analysis was performed and also incurred sample analysis was performed and found accepted. Further, method was revalidated again in presence of 3 metabolites based on the further requirement, and also performed the stability for these 3 metabolites along with the drug. As a part of cross-check between 2 validated methods, like one is the standalone method and another one is a combination method with the 3 proposed metabolite, and few study samples were analyzed as a part of to check the performance of the method between 2. So while comparing the results of cross-check between 2 validated methods, if we can see, then almost all the results are showing the negative bias. So almost 40% samples, minus 40 in average bias was observed. So based on these results, further we plan the investigation to identify the cause of minus 40% bias observed in drug in analyzing study samples between 2 validated methods. One is with metabolite, another one is with without metabolite. So as a part of investigation, we check the COA of reference standards and check whether the COA is acceptable or not. Also check the correction factor as a part of investigation and calculation. Again, we perform the stock weighing activity and check whether there is any issue with the weighing activity associated. Then further prepare the spiking solution. Further check the sample integrity and labeling verification. There is no any sample integrity issue and labeling issue was observed. Also check the sample processing verification, like whatever the equipment used is qualified or not for specifically for this cross-check validation in the real study samples. Then method reproducibility, reliability and ruggedness. Then training and awareness of the analyst. So further we compare both the methods. So there is no any major change in the chromatographic condition between two validated methods, as I mentioned. Only slightly change in the gradient program. So maybe that gradient program doesn't affect the difference in the study samples results. All core experiments in both the validated method found accepted, like precision and accuracy batch size experiment, everything is fine with both the method. Specificity data is okay, matrix effectivity is okay, and process efficiency recovery is okay and found accepted. All stability with and without metabolite are found accepted. So there is no any suspected for a metabolite conversion during the validation or any during the any of the stability. Further, we check the chromatographic condition and separation. If we can see, then all the metabolites are well separated from the drug. So metabolite 1, 2, 3, all three are separated from the drug. So I cannot share the structure due to some confidentiality agreement with the sponsor, but I can just share the metabolite 1 is amide and metabolite 2 and 3 are isomers of each other, that is the glucuronide. So based on this data, we further plan the experiment to evaluate the conversion during the processing or not. So further we plan the experiment. So we check the metabolite conversion sample extraction procedures, then neat sample versus extracted samples. Okay, if we see then in a neat sample solution, we prepare the drug vial, metabolite 2 and metabolite 3 vial. Metabolite 1 is, there's no any interference observed in metabolite 1, so we didn't take that as a part of evaluation. So metabolite 2 and metabolite 3, when we compare the aqueous sample, then there is showing some interference of drug like 3.7% for metabolite 2. So that is only I can say the minimal. But for a metabolite 3, the interference is around 35%. And same, we perform this experiment with the extracted samples to check whether the conversion is coming from any extracted samples or not. But in this case, the neat solution, the extracted samples, the interference of drug from the metabolite is comparable. So based on this data, we can predict that whatever the interference is coming, that is coming from the working standard itself. So further, we, based on this conclusion, we prepare the spiking solution only with the CC by plotting the CC only with the drug, and again perform the cross-check experiment by applying the method 1 by preparing the spiking solution using only drug. So the cross-check between the spiking solution only for drug and the calibration curve standard prepared, and if we can see, then all the results were found well within the acceptance criteria, or I can say 100% samples are within the 20% difference. So based on the overall, the similar percent interference in neat sample and the extracted sample shows that there is no any conversion of metabolite during extraction. Cross-check experiment, more than 40% bias observed between two validated methods due to presence of drug content in one of the metabolite. All the cross-check samples analyzed using combo method were analyzed again with the stand-alone method and found accepted. 100% sample found within acceptance criteria. And this same standalone method we propose to use for a further study sample analysis only for drug, and another validated method has been used only for the exploratory analysis of metabolite. So we divide two methods, and we these two different methods we use for the analysis of metabolite and drug. So I quickly go through the second case studies. So second case study is the method was developed and fully validated by the reference lab, then method was transferred to our biotical lab and fully validated as a part of cross-check. We selected few study samples and perform the in-cut sample analysis as a part of cross-check experiment. Again, we see them. So most of the samples are showing the negative bias. Only the 58.6% samples were found within acceptance criteria, but the remaining all are showing the negative bias. And average negative bias is 22%. So again, to identify the cause of bias observed in analyzed study samples between the reference method and validated method, we check the reference verification, weighing activities, and in each and every stage, we perform the further investigation by selecting the few samples. Then again, check the sample integrity, appearance of the sample, whether the sample is hemolyzed or lipemic. Then sample processing verification, then training and awareness of the analyst. And again, we compare the method, two validated method. There is no any change in the method. everything is same, only few parameter like column brand and is changed, but that should not affect the overall performance of the method. There is no change in the extraction, major change in the extraction procedure. All core experiments were found accepted in both the validated method, and there is no any metabolite suspected based on the metabolite identification data. If we see the chromatogram of the reference method and another validated method, there is no any impurity peaks were found, and also the RT is almost comparable with each other. So extraction method comparison. So I quickly go through the main part of the extraction method. If we see then the spiking solution were prepared and protected for the light, anticoagulant is same, and only the difference in 5% means 2% spiking. Same is also verified as a part of investigation, but there is no any impact with the 5% and 2% with the method. Then sample in both the cases, the extraction method is sample protein precipitation method. If we go down with the protein precipitation, then 300 microliter SN is used in a reference method, while in in-house validated method, we use 0.1% formic acid in acetonitrile. Then again, just with the reference method, they just vertex and inject into the LCMS. And for the in-house validated method, we reconstitute the samples and inject into the LCMS system. So if we can see, then there is two major difference in this method. One is the drying part and another one is the acidic part. So we, as a part of investigation, we process few samples with drying and without drying, but again, the results is same. Negative bias is shown in the study samples. Then we decided to check the condition with the acidic samples. Then we remove the 0.1% acidic formic acid acidic condition from the in-house validated method and perform the cross-check experiment by selecting the few in-cut samples. And if we can see, then after removing the acidic condition from the extraction sample preparation, all the samples were found well within the acceptance criteria. And both the results are comparable with the reference lab method and in-house BA lab method. So overall conclusion, more than 20% bias observed between two methods due to acidic sample during the extraction, which leads to the instability of this NC analyte. Further, all the cross-check samples analyzed using without acidifying condition, and all the results were found well within the acceptance. This same standalone validated method has been used for the estimation. So, thank you. So overall, I can say the learning from the specific these two case studies, and also based on the challenges which we faced during the development of NC method, transfer of the method from one to another lab, and in-cut sample analysis. So as per our experience, I can say always perform the cross-check between two validated method using the real study sample analysis, or while transferring the method within the lab or slightly changing the extraction procedure, always perform the in-cut sample reanalysis to check the reproducibility of the assay. And also recommend upon first time use of any assay, just perform the ISR should be performed, which provide the valuable insight of the methods. Okay, thank you.
Yeah,
Thank you.
Thank you, Vikas, for walking us through a couple of challenging studies in NC bioanalysis. So we'll be having question and answer session also in the panel itself. So right now I'll not take any questions, but we'll call other panelists also, and then we'll have question answers. So before that, Deepak, if you can help me with felicitating Vikas for his contribution. So on behalf of Boston Society, because we are happy that you could contribute, you could devote your time and share with us some of the challenges for a central bioanalytical lab, especially in the NC bioanalysis. So this is a certificate of appreciation for your contribution, and we would have loved to have you in person for this meeting, but with the challenges, we are on the virtual platform. So with this, Deepak, I would like to move forward for getting the fellow panelists and
sure.
The members.
And Srinivas would just take a few seconds moment to thank Vikas for actually accepting our request to reschedule his talk from last week bioanalysis session to today's session because we were doing a poster session. So kind of you, Vikas, we really appreciate for accepting our request and actually taking up your talk today. So thank you for that. And as Srinivas mentioned, we would love to have you for an in-person talk as well. So I think we can now start with the panel discussion and would request all the panelists, Peter, Alex, if you're available, please, if you can switch on your videos, I will then just display you on the screen, and would also invite Prakash and Vinod if you are there. And Peter is also here. And I think I'm not seeing Albert Lee, but maybe he's not there. So I think we can get started. Yeah. So thank you, all of you. And now I would request all the attendees also to feel free to post your questions, and what are questions you already posed. I think the moderators, Srinivas, Prakash, and Vinod will take up those questions with the speakers. So thank you. And over to you, Prakash, Srinivas, and Vinod.
Yeah. Thank you, Deepak. I think this one, we have one question on the chat box that I guess is for Alex. It's from Siang. It's like, is there any universal standard for enrichment or copy number?
Yeah. So is there a standard for copy number? Yes, there's only one form of copy number. So that's just the number of times that the barcode is actually sequenced and observed. For enrichment, though, no, there's not a universal standard there. There's probably some convergence to 2 or 3 different ways, though. But yeah, there's different ways to view what the background is. And so there are some different ways in order to look at the data.
Yeah. Thank you. So I'd leave the rest of the panelists to ask questions, or even the speakers could ask questions to others as well.
Maybe I have, okay, go ahead.
Sorry, Peter, go ahead.
Yeah, I just had maybe a quick follow-up question for Alex. I really enjoyed his talk. So, and you were mentioning at the end, membrane targets are tough. So what kind of strategies can be used to also look for interactions with membrane proteins?
Yeah, so the first technique used to do this was to run the screens against whole cells. And in that way, you could look for binders to targets that are overexpressed on the cell's surface. The tough thing about that is a lot of receptors are hard to overexpress and to keep the cell line stable. Mm-hmm. So because of that, and because getting really high expression levels is a tough thing, those kinds of screens, although they do sometimes yield hits, there's a lot of targets which they just don't work for. So recently there was a group in Hong Kong, Xiaoyu Li, who published a paper where he was able to run such screens against whole cells, but he was able to do it against receptors expressed at the normal levels. And it's a pretty complex process of how he was able to do these screens. But if that technique ends up being robust, then it's something that we could end up seeing applied against a large number of different target types. So yeah, it's still an area which needs work. Probably the most successful version is to solubilize the transmembrane targets, but as you know, producing those kinds of proteins and getting them to a crystallography grade purity is tough. So that's the big challenge there. Once the protein is actually produced, though, running the DEL screen is the same, whether it's a transmembrane or it's not.
Yeah, but I guess it could be possible maybe also when using membrane vesicles, because that's what we typically use also for activity for membrane transporters, and then they are overexpressed. Mm-hmm. So I don't know whether anybody has tried this.
It certainly has been bounced around quite a bit. Probably the challenge there is getting the correct area of the protein exposed to the solution. So if it's completely encircled, then the DEL can't actually reach it. Mm-hmm. And the other bit is to get the to get the concentration at the level which is high enough for the DEL screen to work. So those are the two limitations there that I think the field is still working on. But yeah, there's a lot of groups who specialize in this, and they are trying things this very day, and probably within the next few years, I wouldn't be shocked if there's some sort of a dramatic outcome.
Yeah, sounds interesting. Thank you.
Yeah, no, thanks for the questions.
Yeah, I see some questions here for Vikas. Because I think Srikant, it looks more like a comment. So Srikant mentions that in enantiomers, only one will be pharmacologically active. So in your method, were you able to differentiate? And if so, the second one can be excluded and checked. So any thoughts you have on it?
Yeah, thanks for the nice question. Actually, in this case, there is no any specific confirmation for this NC program, like this metabolite is a pharmacologically active or not active. But based on the metabolite identification data, they selected two positional isomer, just like I mentioned that this is the two positional isomer in terms of glucuronide. So position of that two glucuronide is different. So we just have to ensure that these two metabolite doesn't affect the quantitation of the drugs. That's why we selected two glucuronide as a part of estimation for the combination method as I shown in the case study 1.
Yeah, I see there is another question around that. So Tulsidas is asking for new chemical entities. Is there a requirement to have a fully validated method? Or you can have some parameters to validate the method. So it may not be a fully validated method, but a fit for purpose kind of method. That's his question.
Yeah, as per my understanding, actually we need to from the phase 1 onwards, where we need to check the efficacy and the safety of the patient population. So in that case, the fully validated method is required. But in case of just for a preclinical stage and just for the first information stage, yes, partial method validated method can be explored or can be employed during the study sample analysis. But from that phase 1 onwards, we need to fully validated and regulated bioetical method as according to the guidance.
Okay. There's one question which is like any one of the speakers can touch upon. For low sample volume methods, how the matrix effect can be performed given sample preparation is by protein precipitation method. This is again by Srikant. So he's trying to figure out if you are using the protein precipitation technique for low volume samples, how you address matrix effect.
Basically, the matrix effect, I can say that this is the difference between the ionization in presence of matrix ion and in absence of matrix ions. So that we have to ensure during the development and validation stages itself. Specifically for a protein precipitation case, we need to prepare the aqueous equivalent vial with respect to the extracted samples, or maybe we can use water, and throughout the processing, we just have to extract the drug and post-prike the drug during the protein precipitation and compare the results in absence of matrix ion and in presence of matrix ion. So that's the way to demonstrate the matrix factor, I can say, for specifically for the protein precipitation method.
So thank you, Vikas. So I have, this is Deepak here. I have this question for Alex. So Alex, when you actually go out to small and big pharmas with this product, so do you ever face any challenges in terms of confidentiality concerns by the companies? Because the target and the structure is the whole IP thing the company is running around for any particular indication. So do you face any challenges in convincing them on the confidentiality of this? And the second question would be, once a company takes up a product, a target or a structure from you, then what happens? Does it still, I think it may not be there in your database anymore. It may not be available, or if you can just comment on that. Thank you.
Yeah, so different companies have different ways of viewing this. So some companies refuse to share any targets or target names with anyone outside. In that case, they really do need to use a delight kit because they're not willing to send the target to us and have us actually physically do the screen. Now, if we do use a delight screen for someone, we don't need to know what the target is, so the problem is solved. However, we would know what the list of the potential hits were that were uncovered by the screen, but that's a very long list. It contains hundreds of thousands of potential hit structures, and the company which runs the screen is then free to follow up that very long list in any way that they would see fit. And so we don't know which structures they follow up, and we don't know which hits end up being active. So they feel pretty satisfied with their level of knowledge being high and what we know about their target being very low. Now, as far as the exclusivity on a specific hit, in the case of a self-service kit, both Dell Open and the Delight kits, like I said, we don't know what hits they actually follow up, and we don't offer any kind of an exclusive service there. So, and we don't know what the target was. So there might be two academic groups and they'll both get a Dell Open kit and they could screen against the same exact target and find identical hits. And we wouldn't know that this was going on. In a case where the target is sent to us and we physically do the screen, we might know that we're transferring a certain hit to a pharma client. But as a rule, we don't then remove that from the database. And so a partner that we work with, they have to know that when they run a screen with us and they find a hit, if someone else ran the same exact sort of screen, it's possible that they could have found that same hit. So there is that risk there. You could always pay an extra fee and permanently have that structure then scrubbed from our database, but it's not something that we really advise, because the compound collection has billions of different structures, and really I think that risk is fairly low.
Thank you, Alex. Thank you.
Yeah, sure.
Yeah, there's one question from Dr. Srikanth. It's for Peter. So he writes it for any NC, KQ2 and MDCK permeability will give whether the compound is permeable or not. But even though it's PGP substrate, but still we observe the compound is not reaching the brain. So what are the other transporters that can be taken into consideration to understand and troubleshoot the above mentioned challenge?
Yes, thank you. That's a very relevant question. And so we do know now, and you can find this information also in for instance, the review article that was published in 2013, but also later on, which transporters are the major ones in human brain. We have several efflux transporters. So for the efflux transporters, I would definitely advise to use membrane vesicles to see what is the specific interaction or the specific role affinity, if you want, the activity towards your drug candidates. So nowadays these experiments with membrane vesicles can be done in a fairly high throughput. And then if you have a few uptake transporters, especially the OATP1A2 and also the more widely expressed 2B1. So those can also be evaluated, for instance, in transfected cell lines. If you have specific structures like nucleoside-like structures, you probably can test those transporters too. And then maybe if it's a peptide-like drug also, I think some peptide transporters are there, amino acid transporters at the blood-brain barrier. So it really depends a little bit on the structure then which other transports you want to evaluate, but it's not an infinite list. So we do have an idea there.
Okay, thank you.
Yeah, I also have another question again to Peter, is looking at a lot of computational methods are there available for us to predict DILI and other things. So would you like to comment a little bit about that? Because a lot of there are a lot of softwares and computational methods available for DILI. So would you like to comment on a little bit on that?
Yeah, for DILI, I mean, generally speaking, I think a lot of progress has been made, especially in terms of which in vitro methods can be used. However, like predicting starting from the structure, that's probably still a little bit too early to do that reliably. I mean, if I think about predicting the risk for DILI, I think there are two things. So one thing is based on structure, can we predict interaction with certain targets? But then the question, of course, is in terms of DILI, what are the relevant targets? So there's still a lot of questions unanswered there. We don't know all the underlying mechanisms in terms of the toxicity that some drugs are causing, and then sometimes also drug metabolites. And then the other thing, of course, is the exposure of the liver to drug and metabolites, and especially also the intracellular exposure. And it's also something that within my academic group, we are very much interested in to explore further and combine that that question or tackle that question, especially by PPK modeling in combination with input from in vitro models. Yeah. So some exciting things to happen in the coming years, I'm sure.
Thank you.
Yeah, so just as a just to extend this question to Alex, as you just have heard about the DILI and like metabolism or toxicity related liabilities, do you really think, at least in future, the same product which you had developed for the where we are looking for the affinity, can it be somehow added up with such liabilities around metabolism and toxicity? And maybe that can be also available as a DEL library?
Yeah, so it's not something that I discussed in the presentation that I gave, but we are also working on versions of DEL, which could be used in more of a high throughput screening method. So in this case, the DELs are made on beads, and the beads go into little droplets. And within that droplet, you could run a cellular functional screen or an enzymatic assay. And so in that case, you would have the option of running, let's say, an hepatocyte screen or something along those lines, so that you could determine, are these structures stable, or do they get inside of a cell? Are they active in a cellular assay, and so on. And so it's not exactly clear how long that kind of an assay setup is going to take. Will it be online and something that we can offer to partners maybe next year, or will it take several years in order to develop that? It's not clear yet, but it certainly is work that's underway. These are part of collaborations that I did when I did work at Roche in the past too. So this has been a multi-year long effort, and it is a problem that we would like to solve. It's underway. We have a lab working on it. But yeah, with that said, it's not something that we can offer today. So I didn't bring it up.
Thank you, and good to know. Thank you, Alex. So lastly, I mean, if there are any further questions from panelists, it's fine. Otherwise, would like to request Peter. I think you answered one question from Shrikant earlier in the chat. I think you answered that, but I just would like to request if you can explain it so that the audience, the attendees can also just get a view of that about expression of transporters on different organs, and then based on that, if substrates are decided, or how about the variability there?
Yeah, so indeed, the question there was, so the transporters are showing organ-specific tissue distribution. So we're talking different organs, liver, brain, gut, and so on. And so is there substrate overlap? So the question was, do they have common substrates and inhibitors? Yeah, and the answer really is there. There is maybe even as opposed to the metabolizing enzymes a little bit, there's quite some substrate overlap between different transporters, especially if they're from the same family, especially the OATPs. Say, okay, we know that OATP1, B1, and 1B3, it's mainly in the liver. And then 2B1, for instance, is one that is more widely expressed. But several substrates will be transported by all of these isoforms, or many of these isoforms, And then depending, of course, if you're considering a specific drug candidate, depending on how your drug candidate is depending on some of these transporters and not on the others, then yeah, the PK will be predominantly determined by some organs. I think we're also just starting to recognize the role of transporters in drug distribution and how they affect the volume of distribution and this kind of interactions. So that's also something to be explored further, I would say. But again, there it is good to, at least for the very well-known transporters, to profile your drug candidate very well. So, I mean, if you already know which CYP enzymes, for instance, are involved, or why not know also which transporters are involved to get a complete picture, and then can inform, for instance, then PBPK models to get very good in vivo predictions, and then maybe on the long run, the inter-subject variability that you may have in the disposition profile of a given drug candidate.
Thank you. Thanks, Peter. Thank you for the answer. Any further questions we have?
Yeah, I have a follow-up question to what Deepak asked. When it comes to drug discovery, Peter, there are a lot of different preclinical animals or species that we look at. So what do you think? I think you did talk about the rat MRP3 and the human MRP3 differences, but what do you think would be the right preclinical species of which transporter is more close, which preclinical species transporters are more closer to humans when we actually do the preclinical early drug discovery?
Yeah, that's a very good question. And I mean, in general, and maybe this may sound a bit controversial, but in general, I would say preclinical species are not very good predictors of human PK. So I think if we can use human in vitro systems, that's always preferred. It's of course different for safety evaluation, for toxicity, then you do need the preclinical species, I believe, at least nowadays. So what maybe we still learn then from the PK studies in preclinical species, I think if we do in vitro and in vivo studies in a given preclinical species, this could be the rat or any other preclinical species that is commonly used, then we can learn more about how can you reliably extrapolate from in vitro to in vivo, for instance, and also building PBPK models based on the in vitro data, and then use that knowledge, that experience, I would say, more in a translational way, to make better PBPK models translating from human in vitro systems to human in vivo, where you typically have very limited data than if we're talking about human in vivo. So it's kind of an indirect way, I would say, of informing the in vitro in vivo extrapolation and PBPK modeling. That's how the preclinical species, in my opinion, would fit in.
Okay, all right, thank you.
Thank you. Then I would like to take this opportunity to thank all the three speakers, in fact, Albert as well. So big thank you to Alex, Peter, and Vikas for a wonderful talk and a very interactive session. May now quickly request the moderators for the session for their concluding remarks, starting with Vinod. And while concluding remarks are going on, would launch the poll. So would request all the participants and the panelists to just participate in the poll. Few questions are asked, and so you can submit your responses. It will be on your screen. Thank you. So over to you, Vinod.
Yeah. Thank you, Deepak. So I think today's talks has touched most of the drug discovery area. So maybe from DNA to membrane transportation, their interaction to liver metabolism, and clinical trials, case studies. So I thank all the speakers for sharing their knowledge, their experience, and their dedication in their respective fields. And of course, a very happy birthday to Peter also. So as this will be the last webinar under Discovery and Biotransformation, I take this opportunity to thank all the earlier and future speakers who have made this APA 2021 virtual session a success. Last but not the least, a special thanks to Deepak and all the selfless committee members, and of course, all the attendees for making this happen. Thanks a lot.
Thank you, Vinod. May I now request Prakash for your concluding remarks?
Yeah, so I think Vinod covered pretty much whatever I wanted to say. So first of all, I'd like to thank all the speakers here. A lot of them are either dialing in very early in the morning or very late at night. And also being a Saturday, I think this has been an excellent session with a lot of people coming in pretty much every Saturday, second, third, fourth Saturdays to attend these talks. So I'd like to thank all the speakers from the current webinar as well as the previous webinars, and as well, like where Vinod pointed out, it was like a full spectrum today, starting from screening where Alex talked about, or the hepatocytes, how you do the in vitro assays, little bit more detail about how transporters affect it, and then comes into NCEs analysis. So I think it was kind of a whole spectrum with very different things, and we also got to learn a lot more about each of these things. So I'd like to thank all the speakers here, and again, happy birthday, Peter. Hope you have a great day going forward. Thank you.
Thank you, Prakash. May I
Thank you.
now request Srinivas for your concluding remarks? Srinivas, your line is muted.
Yeah, I echo what Prakash and Vinod had expressed about how this entire deliberations have gone. And I am really happy to see that the last session's talk on NC analysis, how seamlessly got integrated into this session to complete it, taking the NC bioanalysis. And all the speakers really have added a great wealth of information to the audience. And as usual, there are many people like the committee members and organizers have put in a lot of efforts to get experts in the field, even through virtual medium this time. So I'm sure all of us are looking forward to have all these experts coming in physical for maybe the next edition if we have so in physical gathering. And thank you everyone for your contribution to these sessions. And Peter, happy birthday once again.
Thank you. Thank you, Srinivas. And let me just very quickly, in a few seconds, take this opportunity to thank, first of all the three speakers, all three moderators, Prakash, Srinivas, and Vinod, for a wonderful job you have done. And a big thank you to our speakers. And we'll take a moment to extend my gratitude and thanks to the untiring efforts of the organizing committee, because I think I've been blessed to have support from such a very active committee. Such a committee, for the audience, would like to say that we have representation from more than 40 CROs and research companies and academia on our committee, and all of them have been there throughout this. And APA India 2021 was a virtual conference, and we tried this format for the first time, where we split the entire conference into a 6 months long webinar series with 3 Saturdays every month. We were doing this webinar. Today is a second but last 17th webinar, and the committee members and the attendees have been all throughout there. Many of them joining on Zoom and others joining or reviewing the YouTube link. So I would like to thank all of you for your great effort and support. And on a lighter note, I'm glad that now we are reaching to the fag end of it, because I had a lot of trouble at my family also, because 3 Saturdays for last 6 months, starting from June till November. So it was too much. And I think they were really unhappy about it. So I'm glad that now we are reaching towards the end of this webinar series. And hopefully next year, we are going to have an in-person meeting, a 3-day conference as we have been doing, and we will definitely be able to see each other in person next year. So with that, let me thank the moderator as well as speakers, and maybe if speakers would like, they may sign off, and would request the attendees and other panelists to hang on there. We will continue with our webinar poll, the draw which we'll be doing for that. So thank you. Thank you, Alex. Thank you, Peter, and thank you, Vikas.
Thank you. Bye.
Bye.
Thank you.
Bye, guys.
So for the other attendees, the poll is still on. I will keep it on for another 30 seconds. So kindly